afternoon, good morning, good evening, everybody, and thank you for joining us for this session number 11 of this year's World Sepsis Congress. And this session is focused on the research agenda for antimicrobial resistance and sepsis. And my name is Louise Norton-Smith, and I am Director of External Affairs at CARB-X, the Combating Antibiotic Resistant Bacteria Accelerator. So first of all, I would like to thank the organisers and the sponsors of this year's Congress, and I'm delighted to welcome our esteemed panel. We will hear from each of them in turn, leaving a few minutes, hopefully, if we have enough time, at the end of each presentation for a brief question and answer session. So please do submit your question via the chat function. So without further ado, I would like to introduce our first speaker, Dr. Benedikt Huttner. And Benedikt is the Unit Head of Control and Response Strategies in the Division of Antimicrobial Resistance at WHO headquarters in Geneva, leading their work on antimicrobial stewardship and antimicrobial use surveillance. Previously, Benedikt was the team lead of essential medicines in the Division of Access to Medicines and Health Products at WHO, where he led the development of the WHO Aware Antibiotic Book. Benedikt obtained his medical degree in Munich, Germany in 2001, and then moved to Switzerland to train in infectious diseases, where he subsequently worked as a clinician responsible for antimicrobial guidelines and stewardship at Geneva University Hospitals. So Benedikt will present WHO's global research priorities for AMR and sepsis. Over to you, Benedikt.
Thank you very much. Good afternoon from Geneva, everybody. I don't have any conflicts of interest, but I'm a full-time staff member of WHO. As a quick outline of my presentation, I want to first talk about the burden of antimicrobial resistance and a little bit about WHO's response. Then address a global research agenda for AMR focusing on sepsis, and then also mention some examples of WHO-led solutions. You probably will see the similar slides several times. Antimicrobial resistance has a very important impact both on morbidity and mortality. You see that in that Lancet paper published last year, there are about 4.7 million deaths associated with AMR and 1.14 attributable to AMR. And a large part of those deaths are obviously linked to sepsis. And what is the difference between associated and attributable is just what is assumed as a counterfactual. Do you assume you can replace a resistant infection with no infection, or would it be replaced by infection with a susceptible organism? Depending on that, you get different results. Obviously, these results have to be interpreted with some caution, because we know that in the countries that have the highest burden, we also have the least comprehensive and representative data. And that's, by the way, something WHO is working on, looking specifically at bloodstream infections, which is obviously not equivalent to sepsis, but with a significant overlap, to really try to get some representative data on what is the prevalence of AMR and how big is the burden in low middle income countries. So last year has been very important year for AMR. Why? Because there was a UN high-level meeting in New York, and that is nearly 10 years after WHO member states first endorsed the global action plan on AMR. But while we made some progress, there is still need for more progress at the country level. And if you look at the strategic and operational priorities, it's obviously a multi-tonged approach with very important the prevention of infection. If you can prevent infection, if you can prevent sepsis, that usually is much more cost effective than anything that comes afterwards. And we know there are still very important gaps in these areas. As the second priority is having access to both diagnosis and appropriate treatment of infections. Again, crucial for sepsis to have access not only to the diagnostic test, but also to the correct treatments. I'm going to talk about it in a bit more detail later. To have the strategic information, surveillance. You may have heard about the global antimicrobial resistance and use surveillance system. And then obviously the governance and financing, which continue to be a bit of a challenge, and probably even more so now given the recent developments. That's just an overview of the commitment of the UNGA in New York. You don't have to read this in detail. I think the key issue is the aim is to reduce mortality due to AMR by 10% by 2030. There's the idea to update also the global action plan with a stronger One Health focus. You see access is a key component, financing. There are some targets regarding human antibiotic use, prevention, diagnostics. A quite comprehensive set of tasks and challenges we need to overcome to reach that goal. So I was asked to talk specifically about WHO's research agenda, and there has been a research agenda that's been published already some time ago, with also a scientific publication last year that outlines how WHO established the research priorities. So the problem is that there's limited evidence base, especially in low-middle income countries, similar to the surveillance data I mentioned. We need new optimal tools for interventions for AMR prevention, diagnosis, care, and treatment, and how to deliver those. So the idea was to identify and prioritize research topics to inform health policies and evidence-based intervention in the human health sector. So it's mostly focused on bacterial and fungal priority pathogens, but also tuberculosis, with a global geographic context, but including specifically also the few from low-middle income countries. And the time frame that was selected was 2030, which now doesn't really seem very, very far away. So research agenda spanned 4 domains. So on the one hand, they have more the epidemiologic data about burden and drivers of resistance, how interventions are delivered, to improve existing interventions and to discover new interventions. You see that it's about across areas: prevention, diagnosis, treatment, and care, and then more cross-cutting activities. I'm not going to go in much detail because we don't have much time. The prioritization occurred with the help of a number of experts from around the globe, including a presentation from low-middle income countries and specialists that had a more pediatric focus. Again, not to go into detail, but you see that there are 40 research priorities were identified in all these domains. And in the next slide, I chose to highlight some that may relate more specifically also to sepsis. One part is related to diagnosis. We know there's an issue with the lack of affordable and also adequately performing rapid point of care tests to discriminate bacterial versus non-bacterial infections, diagnostic tests for detecting pathogens and have quickly antimicrobial susceptibility results. And a very important issue is, for example, how to use data on antimicrobial use and AMR surveillance to inform guidelines, something we haven't really addressed systematically so far, because we tell countries to collect data. Maybe what's missing is how to use the data. Then with regard to antimicrobial stewardship, I think we know quite a lot, but still, again, evidence gap, many evidence gaps, specifically also for low middle income countries. What are really the best interventions, most cost-effective interventions? How can we optimize empirical antimicrobial treatment for the main infectious syndromes? And then there's some specific pathogens mentioned, and that's the gram-negatives, that extended spectrum beta-lactamase producing and carbapenem resistant Enterobacterales have a very high burden in many settings and can be quite difficult to treat and are often also associated with sepsis. And then a subset of subset population, it's obviously young children and neonates and neonatal sepsis, where the data are even more limited. What is the best empirical antibiotic treatment for gram-negative infections in these patients? Now, I want to mention some WHO-led solutions. You may be familiar with the AWARE framework, so access, watch, and reserve. So since 2017, antibiotics are classified into access, watch, and reserve. Access would be, for example, amoxicillin or amoxicillin clavulanate. They have a lower resistance potential and should be the first and second choice option for many syndromes. And you have the watch antibiotics, which have a higher resistance potential, like ceftriaxone, ciprofloxacin. Very important antibiotic, unfortunately also very often overused or inappropriately used. And then you have the reserve antibiotics, and these are antibiotics like colistin or some of the newer agents like ceftazidime, avibactam, or cefiderocol. They're the last resort option against MDRO. What's very important that is, if an antibiotic, any of the categories is essential, it obviously should be accessible and affordable for all people who need them. And we know that is not the case. And to support the use of the essential antibiotics, we published a few years ago the WHO AWARE antibiotic book, which you can download here. It's available also as a smartphone application. And It's a very comprehensive guidance on infections encountered in primary healthcare, but also in hospitals, including sepsis in adults and neonates, and including chapters on the use of the reserve antibiotics that are on the essential medicines list. In that context, it's important to mention that WHO is currently working on clinical management of sepsis guidelines that obviously will be published in the near future. And the WHO neonatal sepsis guidelines have just been updated and published in December last year. When we talk about the reserve antibiotics, so these are the antibiotics active against the multi-drug resistant organisms, there's actually a need for quite more research. When we look at the data that are available from the randomized controlled trials on which approvals are based, we often have an unrepresentative target population, meaning that patients that really have multi-drug resistant organisms are underrepresented. Children are often lacking, patients from low middle income settings. Often the comparator is maybe not optimal because it's given in a different administration mode, or sometimes you also have comparators where you have a lot of baseline resistance and the trial design is not always optimal. So there are quite a few things where this probably could be improved. I also want to mention the antibacterial agents in clinical and preclinical developments, or the pipeline and the priority pathogen list. So in 2017 and last year, an update, WHO defined the most important pathogens. Initially was mainly intended also for industry to develop, to say which should be priorities for development. You see that the critical group are the gram-negatives, notably carbapenem resistant and third generation cephalosporin resistant Enterobacterales, but also Acinetobacter carbapenem resistant Acinetobacter baumannii. The pipeline report, which you can download, you see also the number of agents that are in the pipeline depending on activity against different groups. We have quite a few antibiotics actually have been approved in recent years, but few are really fulfill many innovation criteria, meaning that they really have a new mechanism of action, or it's a new class with limited cross resistance. So there's still more work to be done. And finally, I want to talk quickly about access. So we know that access to antibiotics is an issue in some setting. It's mostly concerns the reserve antibiotic, but may also concern some access or watch antibiotics. They have different challenges. Again, I'm not going to go into detail, but for the reserve antibiotic, especially for the newer ones, these are low volume products that often have high costs, are patented, are often not even registered in many countries, are unaffordable, and we know that many of them are not listed on national essential medicines lists. So quite difficult for patients in the highest burden countries to have access to them. And it's obviously most crucial for the patients who have severe infections, such as sepsis. Poses a lot of different issues with regard also how do you assure that these antibiotics used appropriately in settings also where you don't have access to adequate diagnostics. So WHO is working with SCART-P to improve access to essential antibiotics and work with countries and partners to look at the market and public health impact, look at the highest potential interventions, reviewing regulatory and policy initiatives, having forecasting models, and then WHO has developed an operational guidance on how to use these new antibiotics to make sure that they're also used appropriately. So in conclusion, I think it seems evident that AMR is associated with a large morbidity and mortality, and a lot of many deaths due to AMR are related to sepsis. And WHO AMR Research Agenda on Human Health outlined numerous areas related to sepsis, from prevention to diagnosis and treatment. And if we want to reach the goal of reducing AMR-related deaths by 10% by 2030, all these different elements need to fall in place, from prevention, access to diagnosis to treatment, science, innovation, governance, and financing. And that's going to be quite a bit challenge, but we need to do our best. So with that, I want to conclude, and thank you for your attention.
Thank you very much, Benedikt. And that was a really comprehensive overview of all of the activity that the WHO is leading on and working with a number of partners. We haven't had any questions through the chat. I think we will move to Kristoffer next, and then if any others come along and you're still with us, Benedikt, I will direct them to you. But thank you very much for that presentation. So we will move to Dr. Kristoffer Strålin, who is a senior consultant in infectious diseases at the Karolinska University Hospital and associate professor at the Karolinska Institute in Stockholm, in Sweden. And Kristoffer's clinical work and his research are focused on sepsis, pneumonia, and COVID-19. And since 2019, he has chaired the development of the National Patient-Centered Clinical Pathway for Sepsis, which is currently being implemented throughout Sweden. He's also leading the development of a digital surveillance system of the population with community onset sepsis in Sweden. Kristoffer is a partner of the European Consortium Impact that works with precision diagnostic tools for rapid stratification of sepsis patients to enable tailored adjuvant therapy with the goal to improve survival from sepsis. And Kristoffer will now talk to us about that early recognition of sepsis as it needs to become an integral part of antimicrobial stewardship programs. So Kristoffer, the floor is yours.
Thank you very much for the invitation, and thank you for the introduction. Yeah, as we just heard, the global burden of antimicrobial resistance has really emerged, and in recent years we've seen more and more deaths from antimicrobial resistant pathogens. As we just heard, this very thorough and important article showed that almost 5 million deaths were attributable or associated with resistance in 2019, and there were about 1.27 million deaths attributable to resistance. And as you see here, you see the different infectious syndromes of these patients, mostly lower respiratory tract infection, bloodstream infection, abdominal infection, etc. And the most common or important pathogens, Escherichia coli, Staphylococcus aureus, Klebsiella pneumonia, and Streptococcus pneumonia. And the UN has stated that antimicrobial resistance is one of the greatest global health threats. And that's also why the UN had this high-level meeting on antimicrobial resistance in September 2024. So antimicrobial resistance is really a global concern, and already 178 countries have developed national action plans for AMR. And one key thing that we just heard about is the antimicrobial stewardship interventions that we can use in clinical practice to try to handle this problem. And the WHO has presented recommendations and, for instance, this practical guide. Antimicrobial stewardship promotes the optimal use of antimicrobial agents, decisions to use them or not to use them, drug choice, dosing, the route of administration and duration of administration. But can antimicrobial stewardship programs be further improved to prevent AMR deaths? This is a very important question, and to answer it, it's very important to know what is the major reason for deaths in bacterial infections. Already more than 100 years ago, Sir William Osler said, except on few occasions, the patient appeared to die from the body's response to infection rather than from it. What it talks about is sepsis. Sepsis is life-threatening organ dysfunction caused by dysregulated host response to an infection. So operationally, sepsis is the combination of infection and organ dysfunction. And with our current sepsis definition, sepsis is defined as a SOFA score increase of 2 or more due to the infection. This is an important paper published 2016 that was also a basis for the definition. It started with 1.3 million persons or patients who were treated in hospitals in the United States and a few in Europe. And among them, 148 1,000 persons, 11% had suspected bacterial infection. Suspected bacterial infection was defined as bacterial culture obtained and antibiotics initially in the care of the patient. And among these patients with suspected bacterial infections, 4.3% died in hospital and 40% had sepsis of all patients with a SOFA score of at least 2. And here you see the mortality in relation to the SOFA score. As you see, these were the patients on non-ICU and these were the ICU patients. And on both different groups, we see very, very clearly that the patients with only 1 or 0 in SOFA score, they did not have sepsis. They had very low mortality rates. But the patient with suspected bacterial infection with a SOFA score of 2 or more, they had a very high mortality rate, as you see. So sepsis is really the major cause of death in bacterial infection. Sepsis is also a time-critical condition. This was shown in, for instance, this American study of 49,000 patients with sepsis in an emergency department in the New York State. And as you see, this on the x-axis, you see the time from admission to antibiotics given. And on the y-axis, you see the in-hospital mortality. So if the patient was given antibiotics within 1 hour, the mortality was about 22.5%. But if it lasted for 12 hours until the patient received antibiotics, the mortality was in mean 27%. And here you see a meta-analysis of sepsis alert systems in emergency departments. And it really showed that sepsis alert systems were associated with reduced risk of mortality with a risk ratio of 0.81, illustrating that sepsis is a time-critical disease. And thus the Surviving Sepsis campaign has recommended for hospitals and healthcare systems, we recommend using a performance improvement program for sepsis, including sepsis screening for acutely ill, high-risk patients, and standard operating procedures for treatment. That was a strong recommendation. The Global Burden of Disease study has proceeded, and this is a paper discussing the antimicrobial resistant deaths from 1990 to 2021. And as you see, the mortality attributable to AMR has increased during this period, and especially it has increased in the population 70 years or older, and also in the population 50 to 69 years. So the authors state that when somebody dies from sepsis, the probability that the organism causing infection is drug resistant is 25% higher in 2019 relative to 1990. And here you see that the forecast in this article, where they forecast what will happen with antimicrobial resistant deaths up to 2050. And you see a real big increase, especially in the patients 70 years older and 50 to 69 years. And most likely a majority of these AMR deaths will be caused by sepsis, which is a time-critical condition. So antimicrobial stewardship should also focus on timing of antibiotics, and thus they should include early sepsis detection in the antimicrobial stewardship programs. We have the WHO sepsis resolution from 2017, and it urges the member states to develop national policy and processes to improve the prevention, diagnosis, and treatment of sepsis, and to continue efforts to combat antimicrobial resistance by promoting judicious use of antimicrobials. So the fight against AMR and sepsis must go hand in hand. So in conclusion, national action plans for AMR have been developed in most countries. Sepsis is the major cause of death in bacterial infections, including infections caused by resistant pathogens. As sepsis is a time-critical condition, national action plans for early detection of sepsis are needed. And the strong interaction between AMR and sepsis motivates that early recognition of sepsis should be an integral part of antimicrobial stewardship programs. Thank you for your attention.
Thank you very much, Kristoffer, and quite a stark reminder about the predictions for the future if we are not addressing this significantly now. So again, we don't seem to be having any questions coming through the chat. I would encourage everybody to place those on the chat there, but nothing for the time being. So I'm going to take facilitator prerogative and just ask, if you don't mind me putting you on the spot, Kristoffer, around on your views on the role of diagnostics in this space and where you see the world going in our need for and rollout of sufficient diagnostics to address the issues of sepsis.
Sepsis diagnostics is important. First of all, sepsis is a clinical diagnosis based on suspicion of infection or verification of infection plus organ dysfunction that we measure with the SOFA score. However, biomarkers could really help this identification and also to aid especially less experienced clinicians to identify sepsis. If we could have a biomarker that we could use routinely, we could really understand early, oh, this must be sepsis, and then we could maybe identify a higher proportion of the patients. But we need better biomarkers that are more specific for sepsis, because we have biomarkers for infection like CRP and procalcitonin, but they do not say if the patient has sepsis or not. So we really need biomarkers that tell something about the dysregulated host response of the patient that really causes the organ dysfunction. I think there is a lot of research around that, using omics techniques to find omics signatures for that. And if we find omics signatures for that, we can try to find easily handled and cheap biomarkers based on those omics signatures that could really be implemented into healthcare.
Great, thank you ever so much. We will now move to Dr. Mo Yin. Thank you, Dr. Strålin, very much. So Dr. Yin is the Deputy Director of the Advanced ID Clinical Trial Network and is an infectious disease physician at the National University Hospital in Singapore. Mo is the lead investigator for the REGARD-VAP trial, which is reducing antibiotic treatment duration for ventilator-associated pneumonia, and the ACORN-HII study, which is a clinically orientated antimicrobial resistance surveillance network for healthcare-associated infections. Mo has designed and conducted large multinational clinical trials focusing on pragmatic solutions for antibiotic-resistant bacterial infections, and she is driven by the idea of using quality clinical research to improve patient outcomes and propel global policies. She has received numerous awards for her achievements in clinical care, research and teaching, and obtained her MBBS from the National University of Singapore and her DPhil from the University of Oxford. So Mo will present to us on the priorities for AMR in hospital-acquired infections. So over to you, Mo.
Thank you very much for the kind introduction, Louise. And it's an honor to be able to present this topic for this audience on the priorities for antimicrobial resistance in hospital-acquired infections. Yeah, so I think the previous speakers have already emphasized on the importance of this topic in terms of burdens of disease around the world. And last year was really a key year for getting political support for this issue. And we've already gone through the benchmarks that we are hoping to set across the world in the next 5 years. And I'll be going through with you from the studies that we have done and also currently conducting the relevance or the pertinence of these benchmarks around especially Asia. So just to highlight or provide a little bit more context on why hospital-acquired infections or healthcare-associated infections are important in antimicrobial resistance. We all know that antimicrobial resistance can be spread throughout the environment, through our food, the water that we drink, and through touching surfaces and interactions with our family members, colleagues, general environments. However, even in Singapore, like a country that's with very strict antibiotic prescription rules and regulations, 1 in 4 population or general public in the community setting actually carry resistance genes in the gut silently. And a lot of us in the audience here may be carrying that, but we are healthy individuals and may not develop infections until we are much older and perhaps contract other diseases like cancer or require transplant in the future. And therefore, the GRAM study that's published since 2019 using global data has highlighted that the highest mortality actually comes from lower respiratory tract infections or pneumonia and bloodstream infections. And these are generally in the vulnerable and critically ill patients. So this really underpins the reason why we embarked on the ACON-HAI study by Advance ID. So Advance ID is a large clinical trial network that's mainly based in Asia that involves over 100 hospitals around the world, mainly in low-middle income settings. And ACON-HAI is our first study embarked by the network, and we focus on 41 hospitals across 19 countries in Asia. And it is a case-based antimicrobial resistance surveillance on hospital-acquired infections. And over a short span of just over 2 years, we were able to enroll more than 10,000 patients. So just to highlight the difference between case-based versus microbiology-based surveillance. Microbiology-based surveillance usually takes a microbiology database collected in the hospital, in the clinics, but on very few occasions, we may know what the patient's outcomes are and also what antibiotics are being prescribed for these patients. So therefore, we focused on case-based surveillance because that gives us a prospective data set. So we know how the patient, when the patient fell sick, what the antibiotics used to treat these patients are, and their outcomes at the end of their hospitalization. And this data set is really important to form the baseline to set targets, as in the UNGA meeting, and also to trend over time to look at various pathogens and disease syndromes. So here I'm showing you very fresh data based on the first 8,000 to 9,000 patients across Asia. And we can see these are the three main WHO priority pathogens, namely carbapenem-resistant Acinetobacter, third-generation cephalosporin-resistant Enterobacteriales, and carbapenem-resistant Enterobacteriales. And we've broken them down into 3 critical syndromes, including ventilator-associated pneumonia, hospital-acquired BSI, bloodstream infection, or healthcare-associated bloodstream infection. And you can see the colors over here represent the proportion of resistance. And you can see critically for carbapenem-resistant Acinetobacter, in many regions in Asia, the proportion of resistance is higher than 90%. And for third-generation cephalosporin-resistant Enterobacteriales, the proportion of resistance is about 40 to 50%. And for carbapenem-resistant Enterobacteriales is also above 40%. And so because we have this prospective study, we have the opportunity to evaluate the antibiotics treatments that's being used for treating these highly resistant organisms. And I highlight here the 2 carbapenem-resistant gram-negatives. Over here, I show Acinetobacter. And on the left, you see empirical antibiotics that's given for these patients. And the majority of them were given carbapenems and polymyxin-based combinations, carbapenem-based combinations. And very often when I speak to colleagues in the US or Europe, they are very often surprised why we are prescribing patients with polymyxins in our region. And this is really solid data to show that the majority of our patients still are very dependent on polymyxins because we do not have access to newer antibiotics. And antibiotics on the right, you see here are the definitive antibiotics that's prescribed for patients after 3 to 5 days from infection onset. Unfortunately, you see here, majority of the patients are still on carbapenems and carbapenem-based combinations, simply because many of the countries in Asia do not even have access to polymyxins. And this is the similar kind of picture for carbapenem-resistant Enterobacteriales. On the left, the empirical antibiotics are mainly polymyxin-based and carbapenem-based. And on the right, again, the majority of the antibiotics, definitive antibiotics given, remain to be carbapenem-based and polymyxin-based. And going back to the first talk, and also the UNGA goal of reducing or of ensuring our antibiotic prescription to be at least 70% in the excess category, which is highlighted by the green color over here, because in the excess group, antibiotics are usually more effective and safer. But you can see over here in the empirical and definitive prescriptions across Asia for hospital-acquired infections, the vast majority of antibiotics prescribed were in the reserve and watch category. And how feasible is it for us to obtain 70%? In 5 years, it will be very challenging. And what about the patient outcomes? And these are the crude all-cause mortality across these infection syndromes. And these infections are including both susceptible and resistant organisms, so we can calculate the attributable mortality. But even when we include the susceptible infections, we see that the mortalities were as high as 41%. And this table shows the attributable mortality, meaning that if we replace these resistant infections like carbapenem resistance and third-generation cephalosporin resistance, what's the proportion of deaths that we can potentially save? And we can see here, if we can adequately treat these infections or prevent these infections, the proportion of mortality that we can save could be quite high as 17% and 13% in these carbapenem-resistant gram-negative. So I've talked about the impact of ACON-HAI, or the potential impact on global policies and setting goals, and also time trends. However, we also wanted to engage very closely with the study sites on a day-to-day basis as ACON-HAI was conducted. And this is an example of a dashboard that can be accessed through the link over here, that we have time trends of various different types of antibiotics used and different types of pathogens within each country and each hospital setting. And I just want to highlight 2 use cases for ACON-HAI study. This is a hospital in Medan, one of the oldest hospitals and the biggest hospitals. And this is Hermawan and Inka here, who are very energetic, and they realized that through doing ACON-HAI, their blood culture contamination rate was 43%. This is really not a surprise for those of us who have worked in Indonesia. And can you imagine, how do we convince the physicians to send a blood culture before antibiotics if 1 in 2 blood cultures are contaminants? Why would we justify the cost? So through doing their local-led initiatives, just over a course of a few months to a year, they were able to reduce the contamination rate to about 10%, 14%. And the second use case is in Calmet Hospital in Phnom Penh in Cambodia. This is a hospital that's tertiary, fairly advanced hospital. And over the last 20 years, they've had 3 versions of infection prevention and control protocols. But never in those 20 years, they managed to set up a team to focus on infection prevention and control. So during the 9 months that they did ACON-HAI, they picked up 268 cases and simply multiplied that by $3,000 per case, which is fairly conservative. And they showed to the administrator that we can potentially save almost $1 million US in Cambodia in this hospital if we were to do infection prevention and control. And almost overnight, in a few months, they set up this infection prevention and control unit, and you can see that their baseline rate was almost 25%. So 1 in 4 patients admitted in this hospital had either a bloodstream infection or ventilator-associated pneumonia. And over a few months, they managed to reduce that rate down to just about 7%, and they managed to show the reduction in cost. So this is just to illustrate that research that can impact global level policies can also have very profound impacts in the local setting. So beyond quantifying disease burden, how can we tackle antibiotic-resistant hospital-acquired infections? And this is a summary slide that highlights the strategy that Advanced ID is adopting. We are building the foundation throughout Asia and globally in our study sites by building a minimum standard in research capabilities through doing observational studies like ACON-HAI. Through ACON-HAI, as we all know, those who are involved in research, we have not only accomplished 10,000 patients, but also the number of contracts that we have to negotiate is well over 120 different contracts with these study sites. And also establishing a database and research teams in each of the sites that facilitates us to go on to do randomized controlled trials. And what we are doing now is include rapid diagnostics to sort of changing the paradigm from phenotypic testing to genotypic testing. We've recently secured a grant to do infection prevention and control in the ICUs. This trial, step wedge randomized controlled trial, will be called Shiny ICU. And also we are adopting a new trial methodology, a design to look at how to better treat carbapenem-resistant gram-negative bacilli. So very quickly, a summary of all these various studies that I have mentioned. The RAPID trial is a phase 4 open-label, multinational, individually randomized superiority trial. So as we all know, if we depend on the conventional microbiology testing, it takes up to 5 days for the reports to be available. But in this trial that we are testing, we are making use of a multiplex PCR platform and working with the hospitals to develop locally adapted antibiotic stewardship to reduce that whole process to potentially hours. And we have a very strict protocol to work very quickly with the lab and the physicians to work on diagnostic stewardship, rapid diagnostic, antibiotic stewardship, and appropriate antibiotic administration, all within 4 hours. And because we have many study sites on board, we are aiming 25 sites in 8 different countries. Since this is the data actually from 2 months ago now, we have already randomized 450 patients over the past year. And our sample size is fairly ambitious at 1,500, which will be the first RCT that's powered for rapid diagnostics that's powered for patient outcomes. So hopefully this will really bring forth very important evidence to break that vicious cycle where there's low demand, high price. So coming on to the treatment trial. So as we all know, carbapenem-resistant gram-negatives are really difficult to treat because each of them have different resistance genes, there's different bacteria causing different syndromes, and there's not many antibiotics available, and those available is very different in terms of availability in different countries, and the patients have different comorbidities, and the physicians have their own preferences. And so a conventional randomized controlled trial with two fixed arms and fixed sample size will not be able to fit this type of context. So this is the practical design proposed by Sarah Walker and now being applied in a neonatal sepsis trial called NeoSep. And we are applying a similar design in adult patients that's making use of personalizable randomization. And this is the key feature of this design, meaning that across all the various arms here, the patients do not need to be eligible for every one of them to enroll. They only need to be eligible for more than one. So as few as 2, they're able to be enrolled into the trial. So that makes it very flexible for enrollment. And we are also making it into a Bayesian adaptive platform trial so that we can drop treatments that are ineffective at interim analysis and add new treatments as they become available. So just of yesterday, we initiated our first study site, and in total, we will have over 40 hospitals in Asia, Europe, and Australia, as well as in the Middle East joining us. And again, we are focusing on hospital-acquired infections that include lower respiratory tract infections and bloodstream infection. And because of the reality on the ground in Asia, we have to focus on polymyxin-based existing combinations because these are the actual antibiotics that's being used on the ground. And also through this trial, we are introducing newer antibiotics like ceftazidime avibactam and making it available throughout the hospitals joining us so that we can, at the end of the day, get a confident ranking of these various antibiotics and not just which one is the best based on an arbitrary p-value. And additionally, we can add factorial randomization on top of the personalizable randomization to add other treatment strategies. So another trial that we are planning for is nebulized colistin for ventilator-associated pneumonia. For this, we are working with Aerogen, an industry partner, so that on top of the intravenous antibiotics, we can also add on a placebo-controlled trial for nebulized antibiotics. So this is my last slide on the access issue. We are also very passionate by this issue that's very, very heartfelt for us because as you have seen in our data, there's an obvious high disease burden in the region, but we do not have access to these newer and safer antibiotics. So for this work, we have done very comprehensive literature review and conducted in-depth interviews with all sorts of stakeholders, as highlighted in the graph on the left here, involving the industry partners, funders, healthcare practitioners, patient advocacy groups, Ministry of Health, health technology assessment agencies, and drug regulators across Asia to find out what are the issues. And we are currently forming an expert panel group that also involves GAP to paint a specific roadmap and how we can bring in these newer antibiotics to the region. So as a summary slide, I think I've emphasized that it is very important to do cohesive surveillance that's ideally case-based across geographical borders to inform treatment guidelines, research priorities, and policies. On the ground, there are locally driven improvements that can be derived from these surveillance studies or even simple research studies that can improve diagnostics and infection prevention and control. And we do need large-scale, cost-efficient, pragmatic, randomized trials, especially in low middle-income settings, to target antimicrobial resistance, especially in hospital-acquired infections. And I cannot emphasize enough that there are low-hanging fruits like prevention, like infection prevention and control, wash and hand hygiene that can really be optimized in low middle-income settings, on top of the newer innovations like diagnostics and newer antibiotics. So in doing all of this, we hope that we are bridging the gap, especially in coming out with good evidence to ensure that our patients access existing newer and safer antibiotics as well as diagnostics. And thank you for your attention.
Thank you so much, Mo. That was fascinating and just shows how much progress that the Advanced ID Network has seen over a relatively short space of time. So very, very exciting. We had one question around carbapenem-resistant Acinetobacter baumannii. What can we do? But I think you've given us a really nice overview of it's a combination of all of those factors, including IPC, of course, and WASH, as well as the work that you're doing on diagnostics and the trials there. So I think unless you had another burning insight to add, I think we will move to Louise's presentation, which I think is very much linked to the work that you've been doing in South and Southeast Asia and Asia Pacific. So it's a really nice segue, I think. So thank you very much, Mo.
Thank you.
I would now like to introduce a fellow Louise. So Dr. Louise Hill is a clinical lecturer in paediatric infectious diseases within the Centre for Neonatal and Paediatric Infection at City St George's, University of London, which is a leading centre for neonatal and paediatric AMR clinical research in Europe. And Louise's research focuses on strategic clinical trials to prevent, prevention and treatment of neonatal infections. Louise is currently a co-investigator for the NeoSep1 trial, which was handily introduced by Mo just now, which is sponsored by GARD-P, which will evaluate new antibiotic combinations for neonates with sepsis. NeoSep1 aims to recruit 3,000 hospital neonates at resource-limited settings across Africa and Asia. So I will pause there. There's lots more about Louise that we don't have time to cover, but is in her biography and potentially in her presentation as well. So over to you, Louise, to talk about NeoSep1.
Hi, and thank you very much for the invitation and for the introduction, and also to Mo Yin for introducing the NeoSep trial. So I'm going to talk about some of the practical considerations in terms of this study. So NeoSEP1 is an open-label randomized controlled trial comparing novel combination and currently used antibiotic regimens for the empiric treatment of neonatal sepsis. And we previously completed a run-in confirmatory pharmacokinetic phase. The study is sponsored by the Global Antibiotic Research and Development Partnership, GARD-P. So why is NeoSEP needed? There are around 2.4 million neonatal deaths every year, with sepsis being one of the leading causes, and the highest burden of disease is seen in low and middle income settings. Antimicrobial resistance disproportionately affects neonates, with around 214,000 neonatal sepsis deaths per year, due to multi-drug resistant organisms, and the highest disability-adjusted life years attributed to infections caused by resistant bacteria are seen in this vulnerable and important population. In terms of causative organisms globally, Enterobacterales and organisms with extended spectrum beta-lactamases now dominate, and we're seeing increasing antimicrobial resistance to the World Health Organization recommended regimens, which have not changed much in the last number of years. So culture positivity rates in neonatal sepsis are often very low, meaning that antibiotic treatment is largely empirical. And so we wanted to focus on empiric antibiotic treatment for neonatal sepsis in this trial. But we first of all needed to understand how antibiotics are currently being prescribed on neonatal units globally. So GARD-P sponsored NEO-OBS, which was a global neonatal sepsis observational study, which recruited over 3,200 infants from 19 hospitals in 11 countries, which were mainly in resource-limited settings. So what NEO-OBS showed was that there's wide variation in antibiotic use, with over 200 different empiric antibiotic regimens started for neonatal sepsis at baseline. There's frequent deviation from the WHO first-line therapy, with a high use of what would usually be classed as second-line antibiotics as first-line treatment. There's frequent escalation and switching of antibiotics that it's demonstrated in this graph in the top right-hand corner, where you can see the initial regimens and then lots of switching and escalation. However, there was suboptimal coverage of the organisms causing neonatal sepsis in the babies with positive blood cultures, despite deviation from the World Health Organization guidelines. So this led to several challenges when designing an empiric antibiotic neonatal sepsis trial, as there's no single accepted standard of care, and there are multiple bacteria etiologies involved in neonatal sepsis with increasing AMR. Also, given the lack of new antibiotics in the pipeline, we wanted to look at the potential of repurposing of off-patent antibiotics with a neonatal license. And so we will be using regimens containing flomoxef and phosphomycin, which both have a described neonatal dose and good coverage of neonatal pathogens, including many ESBLs. And our collaborators have undertaken extensive in vitro work, which has demonstrated that these antibiotics work synergistically when they're used in combination together or in combination with amikacin. And these antibiotics are potentially good candidates as meropenem-sparing regimens. So given the wide variation in antibiotics that are being prescribed for neonatal sepsis, we wanted to test a range of antibiotic regimens in NeoSEP1. However, as NEO-OBS has demonstrated, there is no agreed standard of care, and so using a more traditional trial design, such as a multi-arm, multi-stage trial design, was not going to be able to answer or address our question. So in this study, we are going to be looking at WHO regimens, the other commonly prescribed antibiotic regimens, and then these novel combinations of older antibiotics. NeoSEP1, as mentioned by Mo Yen, is going to use a novel trial design developed by Professor Sarah Walker from the MRC Clinical Trials Unit at UCL, which adopts a personalized selection of regimens and a meta-analytical approach that allows you to rank the antibiotic regimens in terms of those likely to lead to a good clinical outcome for neonates that have particular characteristics, but also helps to avoid the less favorable regimens based based on factors like cost, AMR and toxicity, for example. Given that there are quite a high number of babies have care escalated or the switching of antibiotics, we're going to have a second randomization in the trial, which uses sequential multiple assignment randomization design to allow escalation to second-line therapy. But I'm not going to focus on that today, given the time constraints. So NeoSEP1 plans to enrol 3,000 babies across 20 neonatal units globally and will evaluate relevant locally defined first and second line randomization lists for specific neonatal clinical subgroups. And our primary endpoint is 28-day mortality. And then we have various secondary endpoints based around efficacy and safety. So babies will be screened for the study, and then if eligible, will undergo first-line randomization, and then have daily assessments. If they improve or are cured, then they will be followed up. However, if they deteriorate or don't improve, then they will undergo a second-line randomization where appropriate, again, having daily assessments and follow-up. So in order to be able to do the study, we first of all needed to engage with sites where the trial might be of benefit and ensure wide geographical representation. So we've approached over 40 neonatal units in 19 countries, and we've now selected 20 neonatal units in 11 countries to either be trial sites or reserve sites. So in terms of the implementation of Practical, it can be operationalized in terms of its randomization lists by using a stratification approach, as there are often groups of neonates who share similar clinical characteristics and where empirical antibiotic treatment would be the same. So this allows randomization lists to be pre-agreed with the sites and pre-agreed lists of antibiotics for different groups of babies. So this trial will flexibly compare these multiple different relevant treatment regimens to enable the trial to be run in sites worldwide with variable background rates of pathogens, resistance, and patterns of routine clinical care. But in order to implement it at each of the trial sites, we needed to understand the clinical subgroups of neonates whose characteristics generally dictate which empiric antibiotics are started. So this can include things like whether the babies are inborn or outborn, whether they have early onset sepsis or late onset sepsis. We then needed to understand which antibiotics are currently prescribed for different clinical subgroups at that particular site, and then the general resistance patterns, if known, of the organisms causing sepsis. So if we take a quick example here, site 1 told us that they had these 3 clinical subgroups, so early onset sepsis, late onset sepsis, and hospital-acquired sepsis. They then told us which antibiotics they would usually start empirically for each of these groups of babies. We then showed them the 8 antibiotic regimens that we would like to use in the trial, and we asked them to tell us which of these antibiotic regimens they would be happy to randomize the babies in each of these groups to. So you can see that this then started to generate randomization lists for the trial. And these are different depending on what their usual empiric antibiotics are. For site 2, their clinical subgroups were slightly different, as you can see. And again, these are the antibiotics that they would usually use. And then these are the antibiotics that they were willing to randomize each of these babies to. So you can see that generates quite a number of different randomization lists. And given the number of neonatal units involved in the study, this led to many different combinations of the 8 antibiotic regimens being investigated in the trial. We therefore needed to find some sort of order to reduce the number of antibiotic randomization lists. And so we started to look for common patterns and combinations of antibiotics, which then led us to being able to identify, following quite in-depth conversations with the sites, 8 randomization lists that all of the neonatal units were happy that they could randomize babies in the clinical subgroups relevant to their site to at least one of these lists. So in conclusion, sometimes a novel approach to tackling a complicated problem is needed. And implementation of the Practical trial design has been quite intensive, but it can be done. And what's really nice now is that we know the trial sites extremely well and that they really worry about the babies that they're looking after and if they're providing the best treatment. And the really lovely thing about this particular study design is that it really allows the antibiotic randomization list to be tailored to each site, taking their specific considerations and concerns and needs into account. I'd just like to thank the members of the NearSep1 team, particularly the trial sites, and also the sponsor GuardP and our funders. Happy to take any questions.
Thank you very much, Louise. We don't have any questions on your presentation that have come up so far. There is one for Dr. Yin's presentation that came in just after you started. So if we have time, we will go back to that. And again, if there are any others that come in at the moment. But otherwise, so for the time being, I think we will move to Africa CDC and Dr. Elvis Temfack, who is Head of R&D and Clinical Trials Coordination at the Centre for Science and Innovation at CDC, which is based in Ethiopia. And Elvis is a medical doctor and a researcher with over 15 years of clinical and health research experience and a record of publications in high readership international journals. Before joining Africa CDC, Elvis worked at Epicentre, which is the research department of Médecins Sans Frontières, Doctors Without Borders, as a clinical research coordinator in Malawi, supporting the Ministry of Health in HIV first-line treatment policy change. So Elvis will be presenting on AMR research in Africa and improving sepsis. So over to you, Elvis.
Thank you very much, Louise, for the introduction and for inviting me to be part of this very important discussion. So I'm just going to do a very quick overview on the topic that has been assigned to me. To be able to address this, the outline of our talk would be to do a very brief situational analysis of AMR in Africa, as well as the approach to AMR, and then we look at the component that has to do with the research to improve sepsis. So the first thing I'd like to say is that Africa represents 20% of the world's population and has about 25% of the global burden of disease. And as we all know, 70% of the people living with HIV are in Africa, 95% of malaria deaths are on the continent, and then we have more than 1 out of 4 TB deaths occur on the continent. With respect to antimicrobial resistance, there has been quite a number of publications or commentaries to talk about the resurgence of malaria-associated resistance. I mean, this first one is one of the publications that came from our teams. There are other teams that are talking about Plasmodium falciparum resistance in Ethiopia. And when we look at other infections like HIV, we see that drug resistance is a threat to programmatic approach to such cause of high burden on the continent. And even when this drug resistance threatens the programs and we have the opportunity of changing the regimens, we see that there is also the need to continuously monitor antimicrobial resistance in the patients, as could be seen in this experience that we had in Malawi, where the introduction of dolutegravir for HIV treatment at first line, we had 2 cases of development of dolutegravir resistance in about 6 months period, with no prior history to previous exposure to drugs of the same class. So when we look at some of the major drivers of antimicrobial resistance on the continent, it mostly has to do with overuse and misuse of antimicrobials in humans as well as in food systems. The fact that there are suboptimal vaccination rates would mean that people will develop infections, and the fact that there is also lack of access to high quality antimicrobials would mean people would take the low quality antimicrobials, and the fact that there is lack of diagnostics would mean that a lot of people do over-the-counter treatment. And also, when you look at in the communities, there is lack of inadequate knowledge on AMR. One very important factor which should not be neglected is the environmental contamination from hospital and pharmaceutical effluents. As we all know, in 2016, in the UN discussions in New York, one of the key things that came out was the fact that there is a surge in antimicrobial resistance. And one of the recommendations from this high-level meeting was that there is need for immediate action. And following this, the African Union put together a team to work on an African Union framework for antimicrobial resistance control between 2020 and 2025. And this team was put together in 2018. And they identified key areas where there is need for action, and also how to achieve the objectives of the key areas that needs improvement. One of the key things that needed to be done was to strengthen knowledge through research. And talking about the research bit, from this UN Interagency Coordination Group on AMR, there was a recommendation that there is need for innovation to secure the future through research and development of new antimicrobials, development of diagnostics, vaccines, as well as waste management and other tools and other technologies that were very important to be able to control this really rapidly growing pandemic. And from the African Union side, to address research, there was need for collaboration with partners to develop a list of priority areas for research that can advance public health interventions for AMR control in humans, animals, food, environmental, and plant sector. And there was also the need to encourage public, private, and philanthropic partnership to be able to carry out research for AMR control. However, in a recent report that was published by Africa CDC in November 2024, that was talking about accelerating the continental response to antimicrobial resistance, we observed that from a global perspective, where there was need for multi-stakeholder approach to identify the priority areas for which there is need for research to address antimicrobial resistance, looking at the list, for example, the bacterial priority list for 2024 that was published by WHO, as you can see below the diagram, it shows that most sub-Saharan African leading pathogens that cause death attributable to AMR did not appear on the high or critical list of pathogens of this WHO priority list. Coming back to my initial slide where I was talking about the leading causes of mortality associated with AMR, which involve HIV, malaria, and other infections in sub-Saharan Africa, it became clear that African priorities did not really feature in the global priorities for antimicrobial, for research to control antimicrobial resistance. So from an Africa CDC perspective, we embark on a journey to develop an African health research prioritization framework. And this framework was actually developed because we acknowledge the fact that resources for research are limited or are scarce. And secondly, global priorities may not always align with African priorities. And what are the things that we really are keen to do prioritization on? We are targeting health problems with the potential of driving optimal impact if addressed through research. And our approach is a multi-stakeholder involvement from inception, especially involving people in the communities that are affected by the problems that we really need to prioritize. And this prioritization, which uses an approach which is bottom-up as well as top-down, could be adapted at subnational level, at national level, and regional level, and also at the continental level. And for us to be able to achieve this prioritization at different levels, we need to use an approach which is systematically guided, which would also facilitate aggregation of the different priorities to be able to identify regional as well as continental priorities. And just for information, this framework was validated in November 2024 and launched on the 31st of March and 1st of April 2025. And we have started to roll out this framework. And the objective is to support member states to identify their priorities in different thematic areas using their locally available data. And for this process, we actually identified specific criteria, as you can see on this diagram, where we have the different parts of the diagram that are in red are the different criteria that were considered in the prioritization process as a whole. The basic idea here is when someone is healthy, the health system should do everything to preserve the person in a healthy state such that they don't develop any health condition. And when there are any health conditions that come along, we need to be able to understand the determinants, the risk factors, or the causes, as well as have the means to be able to detect them, know the number of cases, and then have the different measures to be able to manage the cases in order to avoid the most dreaded outcome, which is mortality or the prevalence of disabilities or suffering. So how does this framework apply to antimicrobial resistance? So from a thematic area perspective, this framework would be applied to the different infectious diseases that exist in the member states, where we will use this criteria to, first of all, rank them. And then within each specific infectious disease, if we need to address antimicrobial resistance, there are specific questions in the framework to guide the discussion, to develop research questions that need to be addressed to respond to the gaps. Among which, as concerns antimicrobial resistance, I remember one of the previous speakers mentioned that there is no specific vaccine for antimicrobial resistance, but we could think about, are there better vaccines to prevent the development of the specific disease associated with the pathogen that could lead to antimicrobial resistance? If yes, what is the effectiveness? Is there need for us to develop new vaccines to tackle that specific disease? And as concerns the determinants, we really need to understand the determinants of antimicrobial resistance. In most instances, when people talk about antimicrobial resistance, there is always that tendency to say there is need for antimicrobial stewardship, there is need for sensitization of the communities for proper use of antimicrobials. But there is one other fundamental question from a research perspective. When we look at most of the clinical trials that are done to develop therapeutics in Africa, among the 3% of clinical trials that are done globally on the continent, most of them are phase 3 and phase 4 trials. So what happens with the initial phases of the clinical trials where there is need for determinants of the pharmacodynamics, the pharmacokinetics, which could also be a source of explanation for the antimicrobial resistance, maybe because the dosage of the different antimicrobials that will be used in phase 3 is not adaptable to the diverse populations that we find on the continent. So we really need to understand the key drivers of antimicrobial resistance from a very basic perspective, and we start to see how we can address that using research. Talking about detection, a study that was done by the African Society of Laboratory Medicines in 14 member states showed that among the laboratories that had the capacity to do diagnosis, only about 1.3% were able to do an antimicrobial profile to detect resistant strains of organisms. So we have to start asking ourselves the questions: what are the available tools that we have in Africa to detect AMR? What are their performance? Are they adapted to the context? Do we need to develop new tests? So those are some of the things that we really need to address through research in our research agenda. And then even when we have detected the cases, do we have the right antibiotics to be able to manage them? And do we need to develop new antimicrobials? As we can remember, in the UN recommendation, it is said there is need for innovation to develop new medicines, new diagnostics to be able to address AMR as a pandemic. And then among the patients in whom antimicrobial resistance is detected, do we have the means of management? When we manage them, what is the outcome? Are there specific groups that are more vulnerable to the dreaded outcome of mortality? And last but not least, do we really know the actual numbers? The previous speaker mentioned the fact that in low and middle income countries, there is lack of the capacity to do surveillance to actually know the burden of antimicrobial resistance. So this is the main approach that Africa CDC is using in collaboration with the different stakeholders in the member states, the stakeholders at the global level, to be able to address antimicrobial resistance. So to conclude this overview, we would say that global initiatives for antimicrobial resistance research should consider context-specific priorities, There is an urgent need for R&D to address sepsis from end to end. I was glad when one of the presenters mentioned that we also need to develop surrogates to be able to detect sepsis in cases of antimicrobial resistance. And last but not least, we need a multi-stakeholder involvement to address sepsis to ensure that there are alignment of approaches to prevent, detect, treat, and mitigate negative outcomes associated with AMR. On this note, I would like to thank you very much for your attention. Back to you, Chair.
Thank you so much, Elvis, and for that very timely reminder that, of course, we might have global guidelines, but it's really important that we ensure that those guidelines are adapted to each continent, each country, so that they are the most efficient and effective. So thank you for that. And I'm going to move very swiftly now to Karine so that she has enough time for her presentation. I will be very brief in my introduction. So Dr. Karine Bourgeois is from 144 Pharmaceuticals, and she has 15 years plus experience in R&D of biotech companies. And again, you can find her full biography on the website. So Karine will finish the session with her presentation on advancing therapy in critical care. So Karine, over to you, please.
Hello, so thank you so much for the opportunity to be here and talk to you. So I will then move directly to my topic. So my focus today is the discovery and development of drugs for the treatment of circulatory failure, also known as shock. So this work has been done with more than 25 academic institutions. It's a collaborative work where we actually discovered one of the key pathways leading to shock. So what is then shock? So shock is one of the major causes of death in critical care, with more than 350,000 deaths per year in US alone. And shock can be a result of several different triggers or insults, such as sepsis, myocardial infarction, acute heart failure, among others. So, and it's important to understand that in the critical care setting, some of these triggers or insults all come together in one clinical scenario. For example, a patient has sepsis, then develops septic shock, and because of inflammation and lack of source control, for example, it has a cardiac impairment, and then it develops cardiogenic shock. Therefore, from now on, I will focus my talk on shock as a general syndrome and on a target that we believe drives shock in critical care patients. Shock is one of the deadliest diseases that exists, so the mortality is about 30 to 50%. It's important to understand that shock is not only when your cardiac output is low or when you have vasoplegia due to an infection, and both of these things do lead to low blood pressure or hypotension, but shock is rather a situation when all the organs start failing and eventually shut down, and then the patient dies. So what happens really when a patient is in shock? So this patient is usually in the intensive care unit, and the main treatment options are actually vasopressors, inotropes, and mechanical circulatory support. And these treatments aim to keep the blood pressure at a normal level and favor oxygen delivery to your organs. However, the problem here is most of these therapies are actually toxic per se. For example, high dose of vasopressors are extremely toxic, lead to more inflammation and more downstream problems and future comorbidities. Why is the mortality really that high in shock? Well, it's really an issue because shock is a syndrome. It's not really well defined. So the pathophysiological mechanisms that are playing are not very well known and characterized, and because of that, there's no real effective therapies. So we critically need to better understand shock, the molecular mechanisms driving it, so that we're able to have targeted therapies, precision medicine. In very simple words, there are different stages of shock, and usually the initial and compensatory stages they're not very easy to identify. So usually you don't have very clear symptoms, and the patient is either at work or at home. and there are no real markers or diagnostic tests of pre-shock. When a patient starts showing symptoms, for example, chest pain or a really severe signs of infection, so at this point, this patient is already at the hospital and start having like minimum care. And if this is not contained, this patient migrates or evolves into a situation of irreversible shock. In this situation, the chance of survival are dismal. And that's when all the treatments we have that we call standard of care actually stop working. So what do we do here at 14Force? So 14Force is a company and we are really research driven. So we do a lot of research as a start and we work with tons of academic institutions worldwide. And one of our aims is to identify molecules that we can use as diagnostic tests for early understanding and diagnosis of shock. And for when a patient is already in shock, what we try to do is again identify pathophysiological mechanisms that we can modify, inhibit, and modulate so that we reverse shock and support patient survival. But most important, we don't want a patient just to survive. We want the patient to survive well. And there's pretty much two ways of doing that: diagnosing shock as early as possible. And when the patient is in shock, treat the patient the best you can with the most with the proper therapy that addresses the molecular mechanisms at play. So in the 70s, it was shown that an unknown blood-based molecule would induce myocardial depression and lead to shock. In the early 20s, we found out that DPP3, our target that we are addressing in shock, causes acute cardiac depression. A few years later, we could also show that DPP3 not only affects cardiac function, but also affects vascular function, so the vessels. And in this context, by addressing cardiac, the heart and the vessels, DPP3 could be a common mechanism in mixed cardiogenic vasodilatory shock. What is DPP3 really? So DPP3 stands for dipeptidylpeptidase 3. It's a molecule that is usually inside the cell, and there it's actually a good protein. It's a normal function. The problem starts when DPP3 enters the blood. And in this situation, for example, when a patient is in shock, oxygen is not delivered to the cells and the cells die. And upon cell death, DPP3 is then released in the bloodstream. And there, it perpetuates this negative spiral of shock that could lead to death. So the DPP3 pathway and its prognosis has been published extensively in several peer-reviewed publications, and we validated this in more than 100,000 patients. And our solution to that, when DPP3 is in excess in the blood, it's an antibody therapy that is infused intravenously, and it blocks DPP3 in the blood and reverses shock and improves survival. Do we know that this therapy actually works? Yes, we do. So I will, in 2 minutes, I will explain from animals to humans. So in animals, we have tested DPP3 procesumab in various scenarios. Here on the left panel, you see the heart, an echocardiography of a heart in shock. And on the right video, you see a heart that is completely in its normal functions directly after procesumab was infused. And this can be seen also in the graphic representation on the right. If we look in large animal models of septic shock where cardiac impairment has also been observed, so in the sense of the mixed cardiogenic shock we talked before, procesumab animals also require less vasopressors to keep an adequate blood pressure, had improved respiratory function, and in addition had less inflammation, as you can be seen here by IL-6, and less cardiac damage. All the typical issues you have when a patient is in shock. Do we see this in real patients? Yes, we do. All these findings we have observed in animals, we also see in patients. So we have procesumab has been used as compassionate drug, in a few patients already, and we saw very similar endpoints or very similar results where procesumab improved cardiovascular function, reduced systemic inflammation, improved respiratory function and kidney function in these patients that were in mixed shock at a very advanced stage. They were pretty much before dying. So how do we intend to bring procesumab in the clinic? So we, upon a trigger that can be, like I said, initially a cardiac issue or a severe infection, these patients would come to the hospital, they're admitted. If they start showing symptoms of shock, such as clammy skin, hypotension, these patients are then, they go to the intensive care unit and their blood is then screened for DPP3. If DPP3 is then in the blood, these patients are directly eligible for the treatment of procesumab. So and of course, we hope that by that we prevent the use of toxic drugs that we discussed before. So where are we in the development? So we have shown that procesumab is safe in a phase 1 study in healthy volunteers. We are now doing a proof of concept study in shock patients that is running this year. And in 2026 and 2027, we'll have our pivotal study, which will be running in Europe and US and other parts of the world. And then we intend to bring, hopefully, the drug to patients and save more lives. So I hope I didn't use too much time, and I thank you very much for your attention.
Thank you so much, Karine. We are just 1 minute over, so I think we have done very well, although I'm sorry that we haven't had more time for questions. So I would just like to thank all of our learned panelists today for their excellent presentations. I'm sure everybody will agree that they were very insightful and, if a little petrifying in some cases, but also really heartfelt in terms of the advances that are being made across the world. So thank you again. And I'm sorry if we didn't get to your questions, but thank you for joining us, everyone who is online. And I wish everyone a very interesting and rewarding remainder of the Sepsis Congress. So thank you and goodbye.